Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. Today we are going to talk on biotechnological applications of lipases from paper structure and function of biomolecules to chemical industry is often blamed for our environmental problems. So it is said that the chemical processes, particularly at the large scales in the industry, released lot of pollutants for our environment, and that is why in the last few decades this whole business of bioremediation or waste containment or waste remediation has come up, where the industry is encouraged to minimize the discharge of these polluting polluting byproducts in the environments, in the soil, in the air, and in our water bodies. So because of all these concerns, chemists have long ago started developing strategies for synthesis of compounds and materials which hurt the environment a lot less. So an early outcome of that was the development of a new branch of chemistry which is called green chemistry, and there are some rules of green chemistry, and one of the important notion there is that the catalyst should be used, because when you use a catalyst you derive the faster, more efficient conversions, and that minimizes many things like energy consumption. Enzymes with their high catalytic efficiencies hence obviously have an important place in the development of greener strategies in the production of chemicals. Lipids, particularly the fats and oils, are renewable resources. We have seen that when we discussed that they are basically derived from plants and marine sources, and so these are all renewable resources, and hence their biotransformations, which are basically are done with lipases, become a very important component of this drive towards developing greener technologies, or what are known as sustainable technologies. So it is in that context that lipases, which are so much involved in the biotransformation of lipids, occupy a very important position. Lipases which catalyze these reactions are the subject of these next few modules. So the objective of this particular module are to learn why and how enzymes are used in biotechnology, why lipase is among the most frequently used enzyme in biotechnology, how change in reaction medium leads to the use of lipases as synthesis work, and the use of the glycerol which is obtained as a byproduct in the production of biodiesel. So this is the concept map consists of basically to have a very brief introduction about use of the enzymes in biotechnology, and lipase is an important class based upon that fats and oils are renewable resources, and also the diverse activities of lipases which results in their wider range of applications. So this combination is a very good, very useful synergy. And some of the compounds which are synthesized are esters and detergents, and one of the important ester is of course biodiesel, of which the glycerol is a the byproduct, and we can see towards extreme left hand a sort of a series of reactions which can be done using glycerol. A 2002 estimate records that the number of industrial scale processes in which biocatalysts are used have doubled since 1992. Hydrolases at a level of 40% dominated these applications. Hydrolases, as we know, are the enzymes which hydrolyze carbohydrates, proteins, nucleic acids, and lipids, etc. Along with proteases and carbohydrases, lipases constitute the most important class among hydrolases. As we said, the shift in favor of biocatalysis is due to the fact that biocatalysis follow almost all the principles of green chemistry. The feedstock for growing microorganisms, sugars and amino acids is renewable if we choose them appropriately. Atom economy is maximized. Selectivity makes it possible to not having to use protection and deprotection chemistry. Milder reaction conditions associated with less energy consumption and high catalytic efficiency ensure also low energy consumption. It's an interesting to raise the question that why lipases became the most frequently used enzyme in biotechnology. I think that there are variety of reasons which synergized. To start with, our initial interest in lipases was related to digestion, and in fact that is the lipase, even human lipase, which was studied most extensively. When the biochemistry started, there were couple of processes in which the scientists were most interested. From ancient times, the first of course was fermentation. Second process related to the digestion. Our study of the metabolism started in trying to understand these two processes. Trying to understand digestion was very important, and for obvious reasons, biochemists were interested in finding out that whatever we eat, how does it break down, how does catabolism work, how does it give us energy for our various processes associated with the living organism, how does the anabolism work. So that's where the interest in the enzymes came in, which are involved in the digestion process. Fats and oils were an important constituent, have been an important constituent of the diet, particularly for non-vegetarians, which had lot of fatty stuff in the meats and other similar dietary components. And we already know that lipases are involved in the digestion of these fats and the oils. So lipases as a class of hydrolases along with the proteases came to be investigated very early, became the focus of the attention, and their enzymology was reasonably well worked out ahead of the other enzymes, particularly the non-hydrolase variety. So when we say that the hydrolases became the most frequently used biotechnology, lipases are a part of the hydrolase. But let us see of the as compared to the other hydrolases, why lipases continue to lead this list. Let us look at the another application of lipases which actually came up much early, and has now grown into a tremendous market. Detergents, that's the area. Detergents are used all over the world to clean clothes, utensils, and many surfaces. It was found fairly early that the enzymes as additives in these detergent preparations allow catalytic cleaning. The function of the enzyme is to create fragments of the dirt or stains by hydrolysis so that the other surfactants present in the detergent can take over and do the chemical cleaning. Hence, it is obvious that hydrolases constitute the class of the enzymes which were and are added to the detergents. Proteases were incorporated in detergents about 80 to 90 years ago. Currently, household detergents also contain amylases, cellulases, and lipases. Addition of lipase is necessitated as any oil-based food etc. are major causes of stains on the clothes. It is estimated that the enzymes in detergents constitute more than 50% of the market for industrial enzymes. Hence, for lipases too, use in detergents is one of the major applications of lipases. Cleaning as an activity is associated since ancient times. Around 5000 BC, scouring in river was the way to clean. Around 2100 BC, historical accounts mention that humans living on the banks of the river Tigris used olive oil and wood ash to make soap. It was around 1928 that the sulfation of fatty acids was carried out to obtain synthetic detergents. It was Otto Röhm who in 1931 patented the use of pancreatic enzymes in detergents. Over the next couple of decades, the challenges were to create enzymes which could withstand alkaline pH and other harsh conditions of the washing. Also to create granular detergents so that the enzyme dust does not cause allergy. In the immunology paper, while talking of the hypersensitivity reactions, of which allergy is a very important class, we did mention that the enzymes actually can be very important allergens. So this trying to get them into the granular form was not a irrelevant or unimportant issue. It was actually a very important issue for the people who were marketing these detergents because ultimately their acceptability from the consumers largely dependent upon this, that their detergents did not cause allergy to the people who were using them in their household, shop, etc. It was in 1988 that Novozymes introduced lipolase, a lipase for adding to the detergents. Around 1994, Gist Brockets, another company at that time, introduced Lipomax. Another company called Genecor International introduced Limfast. Both were lipases tailored for incorporating in detergents. Some of these companies have merged, brought up by others. At one time, they were major companies selling industrial enzymes. Novo Sir actually has undergone many changes its nomenclature and continues to be a major supplier of the industrial enzymes, including lipases. However, moving on, while proteases, amylases, and cellulases were blended in detergents much earlier, use of lipases happened much later. To screen the suitable lipase, a SLM test has been developed by the industry. After the ash, products are analyzed by HPLC for checking the efficiency of triolene hydrolysis. This test quickly allows screening of many lipase candidates, as now high throughput screening of the enzymes including lipases is necessary for many other diverse applications. For many other applications, many other high throughput screening tests have been developed. For example, in approaches like protein engineering or directed evolution, one has to have methods which can simultaneously screen the enzyme and the lipase in particular for the desired activity. So, many such tests are available. As the market is huge, lipase engineering by side-rated mutagenesis to improve their wash performance was carried out right from the early years of this application, that is the lipase being having a sizable market value and having a definite applications became the early targets of all these approaches. And as we said that this screening test was helpful, but many more better versions, quicker, more sensitive versions were developed to assess these engineered mutants. Oil and fat processing industry is very old. Many lipases were commercially available for fat splitting and for incorporating in detergents. As these are large scale applications, these enzymes are produced in bulk. For such applications, and given the advantages of bulk production, these industrial level preparations were not as costly and are not as costly as research grade enzymes. That is because the research grade enzymes normally have a higher purity grade, whereas these preparations, what are known as industrial grade enzyme preparations, do not require high purity. As far as enzyme production is concerned, in general, is very high percentage of the production cost is at the downstream processing step. This sometimes can constitute as high as 70 to 80% of the overall production cost. So, as these preparations did not require high purity, these downstream processing costs were not involved, and that cut down on the overall production costs of these industrial enzymes. So, as new applications emerged, these readily available class of enzymes were tried more extensively. For the same reason, proteases and amylases were also readily used extensively in biotechnology, even in its early years. Lipases, however, had two distinctive features because of which they score over these other classes of industrial enzymes. Both features have to do with the nature of the emerging applications. Alex Klibanov's work at MIT in the late 1980s showed convincingly that hydrolases can be used for synthesis if the reaction medium is changed from conventional water rich to low water conditions. So, because of the principle of microscopic reversibility, for hydrolysis, water is a reactant during the hydrolytic activity, but if no water is there, hydrolysis cannot take place. So, in such media, let us say anhydrous octane, a protease can form peptide bond instead of hydrolyzing it. While Klibanov in those early papers used proteases to form esters of amino acids, his lab soon thereafter showed that proteases can indeed synthesize peptides. More laboratories joined in. Notably, early contributions came from two other groups. A Swedish group showed how to optimize the process for peptide synthesis in organic media using proteases. Lipases also turned out to be very stable in organic solvents. A classical work, again by Klibanov's group, showed that a lipase could be heated at 100 degrees centigrade in an anhydrous organic solvent and still retained almost all its activity. Fats and oils constitute very important feedstock for chemical industries. These are also what are known as renewable chemicals. So both fatty acids and glycerol can be the starting point for obtaining many chemicals and materials. Oils and fats of non-edible variety are especially interesting since their use does not deplete our edible oil stocks. Such crops include castor beans, Jatropha curricus, Crambe absimica, and macaw palm, and Licania rigida. In Brazil, many of these have been exploited for biodiesel production. A particular attractive feature of these crops are tolerance to harsh conditions and ability to grow on low fertility soils. In an application like biodiesel production, feedstock cost constitutes about 70 to 88% of the final price. This, of course, is based upon the use of chemical catalysts. In case of use of lipases, the cost of the catalyst is substantially higher. In addition to triglycerides, the unrefined oils of fats also contain mono and diglycerides, free fatty acids, phospholipids, unsaponifiable lipids such as carotenoids, steroids, chlorophyll, terpenes, and even hydrocarbons. In addition to that, the biorefinery concept involves not just the use of fats and oils in the production of chemicals and materials, biofuels, but also use rest of the plant material for obtaining value-added product. Extensive enzymatic as well as non-enzymatic routes for valorization of the waste for oleochemical industries have been developed. The fatty acids present in the triglycerides form oil crops. They affect the properties of the biodiesel ultimately produced. The fatty acids with higher carbon chain leads to the biofuel with higher cloud point and cold filter plugging point. Biodiesel produced from such fatty acids is not suitable for use in places where ambient temperature is low. Similarly, a biofuel with better combustion quality, that is a cetane number, is produced from saturated fatty acids or mu form. The presence of many double bonds in the fatty acid chain will also make biofuel more susceptible to oxidation, degradation, and polymerization. In general, biodiesel from fatty acids such as oleic acid or racinoleic acid is reported to produce high quality biodiesel. Waste fat from animal sources has also been used for various biotechnological applications, including biodiesel production. Rendering consists of heating carcasses with water at high temperatures or even steam to release fat. One key parameter in the biodiesel production is the presence of free fatty acids. The alkali catalyst most frequently used in biodiesel production produces soap with the free fatty acids. Use of refining to remove free fatty acids increases the production cost. Biodiesel is an alkyl ester of long chain fatty acids. The alkyl group is generally from short chain alcohols such as methyl alcohol and ethyl alcohol. The alkyl esters of higher chain alcohols are also important as these can be used as a biolubricants. The esterification of fatty acids by short chain alcohols, as mentioned earlier, is most frequently carried out by sodium hydroxide or potassium hydroxide at moderately high temperatures. This consumes energy, corrodes vessels, those production vessels, and produces soap from those free fatty acids. Apart from biodiesel and biolubricants, esters of different acids and alcohols are also used as flavors and fragrances. We will discuss this aspect more in the next module when we discuss organic synthesis with the help of lipases. As we mentioned in the earlier module, lipases show broad specificity. Part of that is that these enzymes can also act as esterases. So the reverse synthesis can produce esters. Esters like ascorbyl palmitate have been produced by lipases. Ascorbyl palmitate is the fat soluble antioxidant which we mentioned earlier. Acylated sugars like palmitoyl glucose etc. are amphiphilic and hence constitute important food grade biosurfactants. Use of lipases in producing these esters, classes of substances like biodiesel, biolubricants, fragrances and flavors and biosurfactants, allows the use of milder conditions in terms of temperature and pressure. For example, in China, a plant producing biodiesel from soybean with the use of a lipase has been operating for many years, and some more similar plants are being commissioned elsewhere. One advantage of using lipases for producing biodiesel is that the byproduct glycerol is of cleaner quality and does not require extensive downstream processing. This makes glycerol obtained through this lipase-based technology useful for many more sectors like pharmaceuticals and cosmetics. Traditional uses of glycerol include its use in synthesis of trinitroglycerine, alkyl resins and polyurethanes. Its use in alkyl resins and regenerated celluloses is a softener. Surface coatings and paints use this as an ingredient as it imparts toughness. A special quality of paper use glycerol. As a food additive, it is used as an additive commercially known as E422. Its use prevents baked goods from drying. It is also used in cough syrups and expectorants. During World War, glycerol was a national defense priority in Europe as soap industry was not producing enough of it to meet the demand as a precursor for dynamite. To meet this challenge, production routes for making glycerol from epichlorohydrin was established. Epichlorohydrin is made from propylene and thus glycerol production became dependent upon petroleum industry. Advent of biodiesel changed this picture drastically. In 1992, about 600,000 tons of glycerol was available as a byproduct of biodiesel synthesis. At present, this is the only source of glycerol. This large amount of glycerol availability has led to extensive research and development efforts in searching for alternative and new applications of glycerol. These applications are independent of the route by which biodiesel production is carried out. However, lipase-based route produces better quality glycerol. Glycerol has been found to be a useful solvent for carrying out many organic reactions. Some other solvents derived from glycerol have been developed. Even for enzymatic reactions, glycerol has been explored as a greener solvent with very low consumption of energy. A small amount of carbon monoxide is also produced in this process based upon favorable water-gas shift thermodynamics. Both hydrogen and methanol are considered cleaner fuels. Syngas can be used as a source of fuels and chemicals. 1,2-propanediol is another very important chemical intermediate. It is obtained from petroleum sources. Obtaining this important commodity chemical at commercial level by reduction of glycerol has been a great achievement. This route is inexpensive and utilizes copper chromite as catalyst at 200 degrees centigrade. Davy process technology has developed as alternative process for achieving the same. 1,3-propanediol can be now obtained using glycerol as a feedstock in the fermentation processes using bacterial strains from Citrobacter, Enterobacter, Quibacilla and Lactobacillus. A new antifreeze mixture of 70% PDO and 30% glycerol has been produced using biodiesel production facilities. 1,3-dichloro-2-propanol can be obtained by halogenation of glycerol and can be converted to epichlorohydrin by dehydrochlorination. Epoxy resins are used in marine, automotive, housing and canning industries and have over billion US dollars market. Epichlorohydrin is also used as a precursor for 1,chloro-2,3-propanediol. This compound is used by pharma industries in cough mixtures. In 2007, Solvay has started a 10,000-ton plant facility for producing epichlorohydrin from glycerol. This glycerol was sourced from a French biodiesel producing company. Dow Chemical similarly has a 150,000 plant in China for producing epichlorohydrin from glycerol obtained from small local producers. These producers obtain their glycerol from biodiesel production from mostly rapeseed oil and palm oil. So in this module we have learned that how enzymes are used in biotechnology. Lipases is most frequently used enzyme in biotechnology, production of esters including biodiesel, by medium engineering of lipases catalyzed reactions, and application of the glycerol produced as a byproduct of biodiesel. Biorefineries are an important part of the global efforts to reduce dependence upon depleting petroleum resources. Simultaneously, these represents a shift from bioremediation to green processes which produce less waste. In fact, the strategy is to abolish the concept of the waste and establish valorization processes. So, whole lot of products have come out based upon glycerol and lot of new chemistry and biological transformations have been developed. Lipase based process gives better glycerol. Thank you.